Welcome, student. In this sessions, we are going to discuss the alternative to practical exam solution. IGCSE Biology Alternative to Practical Paper 6 consists of the main component, data calculations and analysis, plotting the graph, drawing the diagrams and magnifications, and planning and investigations. In these sessions, we are going to focus on the data calculations and analysis. Then the first question, we will carefully read the main statement of investigation. For example, a student investigates the effect of temperature on the water uptake in the celery stalk. So after reading carefully these investigations, we will comprehend and rephrase it. For example, the different temperatures effect on the water uptake in the celery stalk. And while reading the investigations, we will point out the following points. Number one, the dependent and independent variables. Number two, the variable remain constant or the same. And number three, the controlled variables. And number four, the potential source of error. In these investigations, we have the celery stalk and the red stain of equal volume with warm and cold water. We will cut the celery stalk of equal size and the volume and put into the red stain for 10 minutes. And after 10 minutes, we will remove the celery stalk and cut into the 5 millimeter section and observe the presence of red stain in xylem tissues. And we will repeat cutting until the red stain is present. And then we will compare the result of cold water with warm water celery stalk. In this investigation, the step number one, the one celery stalk was placed into the beaker containing warm red stain. That's our one treatment. While another will put into the beaker containing cold red stain. That's the treatment. And both will stay for the 10 minutes. And after 10 minutes, we will remove one and cut into the 5 millimeter section and inspect the presence of red stain. And we will repeat cutting until the red stain completely disappears. And we will repeat the same procedure with the two different, the celery stalk with the cool water and celery stalk with the warm water. And then we will compare the results. Now the first question possible related to this investigation, the state one safety precautions that should be taken while carrying out this method. So the one safety, any possible, you can write any one statement. Use the gloves and the goggles to avoid stain on the body. Or use the scalpel by handles to avoid the cut. And don't use excessive force or the stabbing motions. So any one statement you can write for securing 1 mark. Second question is possible related to the data calculations. Prepare the table and record your results in your table. Your table should contain the following information. Number one, the temperature, cool or warm, of the red stain, and the number of the sections that were stained in the 10 minutes, and the total distance moved by the red stain in the 10 minutes. Now, the number of the 5 millimeter sections were cut from the celery stalk in the warm red stain. They were 11. While number of the 5 millimeter sections cut from the celery stalk of the cool red stain, they were 5. Now this is alternative to practical. The examiner will give you the calculated data and you're supposed to present this calculated data in the given table. For the marking scheme of the given table, the first mark is you are supposed to draw the table with the suitable columns and the rows. For example, the one column we need for the temperature and the second we need the number of the sections for the red stain, and the third column we need that for calculation of total distance covered. So the 1 mark is the equal column or suitable column and the rows. For the second mark, you are supposed to write the headings. For example, the first is the temperature. The second column, the number of 5 millimeter sections stained in 10 minutes. In the third column, the temperature distance moved in millimeter but in 10 minutes. So that's our second key point. Now the third key point, you are supposed to put the two correct distance determined. For example, the one is the warm and second is the cold. So in the warm water, that the total number of pieces are 11. So these 11 pieces, so each piece contains 5 millimeter. So 11 multiply 5 is equal to 55. While the cold water that contains 5, so 5 multiply 5, so that is equal to 25. So in this, the plotting the table, putting the tables or putting the values in the table, you are supposed to be careful that don't put any two values in a one box, and number two, that don't put any unit into the box. Otherwise, this will consider the deduction of marks. Your unit should be present in the heading, and heading should have with the proper unit. So for the mark distribution, number 1 mark is the correct columns. Number 2 mark is for the correct heading with suitable units. For example, 5 millimeter in 10 minutes. So number of section in a 5 millimeter in a 10 minutes. So these are the heading with correct units. And number 3rd mark is the correct, the plotting the data. For example, the 11 and multiply 5 is equal to the 55. So this will give you the number 3rd marks. So this is the total 3 mark questions. I'm heading to the second question. The next is the question number 3. Use the data to calculate the rate of movement of red stain in the celery stalk at each temperature, and space for working will be given in your paper. So we studied the data we have calculated for warm and cold. So we are supposed to put the warm water, how much distance is covered by the warm water. So the formula is the velocity, total velocity is equal to the total distance divided by the total time. Now the total distance we will get from the question number 2, the 55 millimeter divided by the 10 minutes is equal to the 5.5 millimeter per second. This will be our 5.5. While the second, the velocity is equal to 25 millimeter divided by the total time 10 minutes, and that will be the 2.5 millimeter per minute, and the answer will be 2.5. So in this way, you are supposed to calculate your data. You have to show your calculations to your examiner and put the correct value in the blank spaces. We are moving to the next question, that is the state or conclusions for the results. For example, in the question number 3, we collected the results, and results are the warm water 5.5 millimeter per minute, and in the cool water 2.5 millimeter per minute. So the state the results for 1 mark. So the result is the velocity increase by increase of temperature. So you can use alternative words. So by increasing the temperature, the velocity in the celery stalk of water will increase. Now we are moving toward the type of variables. Before going to the type of variable, we will reinforce the idea about independent and the dependent variables. The independent variables in the control of investigator mean the investigator set the values. They do not depend on other variables, and they are represented along x-axis in the graph. For example, in this experiment, we have the type of plant or concentration of the stain, and the type of stain, and the volume of the stain, and temperature in the stain, and the size of cut pieces, and the temperature, while the dependent variables include the result of experiment and dependent variable. So they are the dependent on independent variables which are set by the investigator, and these are basically the result of your experiment, and we represent always these dependent variable along y-axis of the graph. Now, for example, the velocity of the stain in the celery stalk, these are the dependent variables. Now the question number 4, state the two variables which were kept constant in this investigations. So what are the different two variables which we kept constant which can affect our results? For example, the length and width and the surface area or the size of the celery stalk. So these are the two variables we will keep constant. And the size and the width of the cut pieces or the type of the plant or the time of a sections or concentration of the sections. So these are the different variables which can affect our results, and we will keep them constant. So number 5 question. So one way of improving method is used to investigate, like we can repeat the investigations while identify two other sources of error in this investigation, and for each error suggest an improvement to minimize the effect of the error. Now in this investigation question, there is a 4-mark question, so we are supposed to mention the two errors for 2 marks, and we supposed to present one improvement suggestion, so to improve the relevant the error. For example, the number 1 is the xylem tissue may be different with a different size of stalks. So for example, if we are getting the xylem stalks, and if the one xylem stalk is the with a high, the large size, or width is a larger or high mass, so it may have more vascular bundle as compared to the thinner one. So this is the error. So alternative words you can allow, the xylem tissues may be the different with a different size of the stalk. The improvement possible, select the stalk of the same size or same diameter. So that's improvement. The second error, the sections were too big to give precise results. So the improvement means the section should be cut more thinner to increase precision in your calculations. Okay, students, so we have completed the first questions in the data calculation and analysis. Hope you got the idea. Your comment will be well appreciated in the comment box, and that will be more helpful to improving further videos. Thank you very much for watching this video.